Hello everyone, this is Divya from Biotechnica, and today I am here to give you some insights regarding cancer biology. What are the mechanisms and therapeutics used to cure this cancer? So, this particular topic is important when we look into CSIR NET Life Science syllabus. And at the same time, it is also important for other competitive exams like DBT, ICMR, even GATE. So this is a very common topic when we go for competitive exams. So the agenda for today's session is we will be starting with the comparison of a normal cell to that of a cancer cell. What are the type of genes that cause cancer? How a proto-oncogene is converted into an oncogene, and how a tumor suppressor gene is converted into an oncogene. Then the therapeutics related to it. So let's begin with the session now. As I mentioned, the first will be comparison of normal cell to that of a cancer cell. So normal cells are small and they have uniformly shaped nuclei. They will be having large cytoplasmic volume, whereas cancer cells are large, variable shaped nuclei and their cytoplasmic volume is relatively smaller. If you look into the size and shape of this normal cells, they have a uniformity. They are arranged into discrete tissues, whereas cancer cells, there is variation in size and shape of the cell, and there is disorganized arrangement of cells. Normal cell will be possessing differentiated cell structures and they have normal presentation of cell surface markers. But cancer cell, they do not have any specialized feature, and also the expression of cell surface markers are more or elevated. Normal cells will have lower levels of dividing cells and their tissues are clearly demarcated. That means they have defined boundaries beyond which it will not go. But cancer cells are large number of dividing cells with poorly defined tumor boundaries. So now you got a clarity regarding the comparison of how a normal cell is different from a cancer cell. Now let's look on to the genes that are involved or that are responsible for causing cancer. So we have three categories of genes. Proto-oncogenes, basically they promote cell survival and proliferation. Examples of such proto-oncogenes include anti-apoptotic proteins, proteins involved in signal transduction pathways, that is the specifically the proliferation related signaling or the transcription factors that are involved in the signaling. And such a proto-oncogene will undergo gain of function mutation, genetically dominant, getting converted into an oncogene. Its origin could be maybe a point mutation or a chromosomal translocation or gene amplification. Second is tumor suppressor genes. As the name indicates, they inhibit the cell survival or proliferation. That means unnecessary genes. Examples includes apoptosis promoting genes, inhibitors of cell cycle progression, checkpoint pathway proteins, etc. And these genes will undergo loss of function mutation in order to convert into oncogenes. So mutations are genetically recessive and they arise by deletion, point mutation or maybe methylation. Third category of genes are genome maintenance genes, whose normal function is repair or preventing any sort of DNA damage. So basically all the repair enzymes comes under the example category of genome maintenance genes. They also, the effect of mutation will be loss of function mutation, genetically recessive, may be arised by deletion, point mutation or methylation. So there are a number of genes, whether we categorize as proto-oncogenes or tumor suppressor genes. So it is not possible to explain each of the genes, their original function and their mutated function. But yes, I have come up with some examples from both the categories of genes. So first we have how the proto-oncogenes are converted into oncogenes. Three common mechanisms that are involved is, as I told you, a single nucleotide mutation can make it constitutively active. Gene amplification. Amplification indicates increase in the gene expression, protein production. So, proteins will be produced in excess. Chromosomal rearrangement, that means sometimes the chromosomal translocation takes place and the position where it gets translocated could be a strong enhancer. So, in that case what happens? Again there will be increase in the protein expression. Now let's check out some examples of these oncogenes. How are they working? So oncogenes could be the one that encode growth factors or their receptors. For example, cancer-causing sarcoma virus. They contain an oncogene known as CIS, which is derived from PDGF, a protein present in the human blood. So the cultured cells that are transformed with this virus, that means the sarcoma virus, what happened? They secreted large amount of this growth factor in the medium that leads to uncontrolled proliferation of cells. So the overexpression of this PDGF has been implicated in the development of brain tumors. Similarly, we have avian erythroblastosis virus. Now they are found to carry an oncogene known as ERBB, which encodes EGF receptor. Now again this is a growth factor receptor. So the altered version of the receptor stimulates the cell constitutively whether the growth factor is present or absent in the medium. So again uncontrolled proliferation of cells. Second example of oncogenes are those that encode cytoplasmic protein kinases. We all are aware of RAF as a proto-oncogene, the most common protein or the proto-oncogene involved in MAP cascade, or I can say RAS pathway, right? So this protein is having a regulatory domain and a protein kinase domain. So unless and until it is activated, the kinase domain will remain inactive. So what happens if the regulatory domain is replaced with partially deleted viral GAG sequences? This results in activation of protein kinase domain, that means constitutively active, resulting in cell transformation. Third, oncogenes that encode transcription factors. So as we all know about MYC as a transcription factor, what is their function? They regulate the expression of number of proteins, even the non-coding RNAs. For example, we have rRNA, tRNA, even microRNAs that are involved in cell growth and proliferation. So this proto-oncogene is found to be altered in most of the human cancers, and their mode of action could be either it will be gene amplification or it will be chromosomal translocation. So in this image we can see there is a translocation event occurring between chromosome number 8 and chromosome number 14. So this particular part of the gene is translocated over here. And we know that immunoglobulin heavy chain gene is responsible for continuous gene expression. So when the c-MYC gene translocated to this position, what happens? There is increased rate of gene expression over there. Now this is found. This disease or condition is known as Burkitt lymphoma. Next we have the oncogenes that are encoding the proteins affecting epigenetic state of chromatin. Epigenetic means there is some temporary changes or the post-translational modifications occurs in the chromatin level. So it could be either in the DNA or it could be in the histones. That means we have phosphorylation, methylation, acetylation, all that are involved in regulating the gene expression. So to carry out this process, we have a number of enzymes involved. DNA methyltransferase, histone acetylase, deacetylase, histone demethylase, methyltransferase, etc. So all these proteins are present within the chromatin remodeling complexes. So mutation in any of these genes can promote tumorigenesis either by increasing or decreasing the rate of gene transcription. So we have one example for that, AML, that is acute myeloid leukemia. This is characterized by recurrent mutations in DNA methyltransferase 3A enzyme. The original function of this enzyme is maintaining the methylation pattern during the DNA replication. So if methylation pattern is affected, overall replication will be affected. Fifth one is the oncogenes that encode metabolic enzymes. So we have TCA cycle enzymes like isocitrate dehydrogenase, normally responsible for the conversion of isocitrate to alpha ketoglutarate. But if mutated, it gives rise to an abnormal metabolite known as 2-hydroxyglutarate, which gets accumulated at higher levels in the tumor. And such accumulation was seen in patients with glioblastoma as well as acute myeloid leukemia. So these high levels of 2-HG have an impact on a number of processes that includes histone demethylation, DNA methylation. That means basically somewhere they are affecting the epigenetic state. Other than that, as I mentioned previously, point mutation. So if you see, there is a single nucleotide change that has replaced glycine to valine, and this kind of mutations are detected in bladder carcinoma. It is a RAS-H oncogene wherein this point mutation occurred. In some cases, we will find that a large group of oncogenes encoding growth factor receptors, and most of which are protein tyrosine kinases. So we know that platelet-derived growth factor, upon binding to the receptor, the kinase will be active. Now what happens if the extracellular domain part of the receptor is replaced with a TEL as a transcription factor? This results in constitutively active kinase. So what happens? Uncontrolled proliferation of cells. So that's about some examples from oncogenes. Similarly, we have tumor suppressor genes. As you can see, we have APC, BRCA1 gene, the mismatch repair genes, TP53, PTEN gene, RB gene. All these are examples of tumor suppressor genes. So let's check out how exactly it works. So BRCA1 and BRCA2, normally they are present in both males and females, and they are involved in DNA repair process, contributing to genomic stability. But if it gets mutated, it results in ovarian cancer or breast cancer in female. When it comes to male, they result in prostate cancer. P53 as a tumor suppressor gene is considered to be the guardian angel of the genome, because if any damage happens, this is the first gene to sense and recognize it and act accordingly. So mutations can prevent P53 from functioning, and if this happens, what happens? Damage will neither be detected nor be repaired. So what happens? Cells will keep on dividing with the damaged part of the DNA, making it most cancerous. So mutations in this gene are found to be in 50% of cancers. See, this is the normal functioning of P53 gene, whether we have a DNA damage, cell cycle abnormalities or hypoxia. Either it can cause cell cycle arrest, leading to recruitment of repair machineries, then restore the cell cycle. Otherwise, directly activate the apoptosis if the damage is too severe. But if we have a mutated form of p53, no matter what is the condition, cell cycle proceeds, making it cancerous. Let's take an example of this. Suppose we have a DNA damage, double-stranded DNA damage. So, if tumor suppressor genes are good, that means if they are well functioning, then they will help in the DNA repair. But if these two genes are mutated, in that case what happens? A checkpoint activation happens, wherein ATM kinase will be activated, which will go and phosphorylate the p53 gene. So once your p53 is phosphorylated, it results in its activation. Once it is activated, as I mentioned before, either it will halt the cell cycle by increasing the levels of p21. If the damage is severe, it will increase the pro-apoptotic proteins like BAX, inducing apoptosis. Now, what is this MDM2? MDM2 is a negative regulator of p53 gene. So, normally it keeps the p53 at low level in the cell. When required, then only it will be activated. Next, we have p10 gene. So, p10 is a common protein that is involved in the signaling process. In fact, I must say it results in termination of a signaling process. Now, this is related to PKB or AKT pathway. So, PI3 kinase is responsible for transferring the phosphate to PIP2 so that we will get a triphosphate here, phosphatidyl inositol triphosphate, which in turn activates the protein kinase B, resulting in cell proliferation and survival. So, after a period of time, P10 comes and it breaks this PIP3 back to PIP2. So, what happens? Termination happens. But if this P10 gene is mutated, in that case, it will be control cell proliferation and cell survival. Next, we have the restriction point control. So, P16, if they are combined with the cell cycle protein kinase, then it is considered to be inactive. But if they get a signal and that signal removes the P16 from this complex, kinase becomes activated. So once the kinase is activated, it will phosphorylate the RB gene, releasing E2F as the transcription factor. So they will carry out the transcription of necessary proteins that are required for DNA synthesis. So, to summarize, what I can say is that, remember, single allelic mutation converts proto-oncogene to oncogene. It is a dominant cum gain of function mutation. On the other hand, tumor suppressor genes require two allelic mutation. Then only it gets converted into an oncogene. That's why it is a recessive mutation and occurs due to loss of function mutation. So remember these 2 points, important from examination point of view. Then we have many biological agents that can cause cancer. For example, the Burkitt lymphoma, which is caused due to MYC translocation, can also be caused by Epstein-Barr virus. Hepatocellular carcinoma caused by hepatitis B and C virus. Kaposi sarcoma resulted from HIV. HPV causes cervical cancer. Similarly, human T-cell lymphotrophic virus causes adult T-cell leukemia and lymphoma. Now moving on to the last part of this session, that is therapeutics. So different types of cancer treatments are available here. So the first one, as you can see, we have the most common, that is surgery. So surgery is effective in the treatment of primary tumors. Okay, so when used as a single treatment, it can cure more patients. But patients with cancers that cannot be cured by surgery must go for other treatments. Radiation therapy. So basically gamma rays, or we can go for X-rays, particle beam is commonly used. Chemotherapy, the use of chemicals. Immunotherapy, the use of immune cells. Hormonal therapy, that is certain hormones if produced in excess like estrogen and all, it can result in cancer. Targeted therapy, that is you are going for a specific target cell organ for treatment. And the last one is the stem cell therapy. Stem cell therapy, in fact, I must say that it is a rapidly evolving field with significant potential for improving cancer treatment outcomes. So they focuses on, as the name indicates, stem cells to repair, regenerate, or even replace the damaged tissues. It enhances the immune response against cancer and improves the efficacy of existing cancer treatments. So there are various types of stem cells that can be used for this. We have hematopoietic stem cells, mesenchymal stem cells, induced pluripotent stem cells. Then we have also cancer stem cells. Okay. Next, let's have a look into chemotherapy. So as the name indicates, as I told you, we are using drugs or chemicals to kill or inhibit the growth of cancer cells. So chemotherapy is used to treat cancer. We call it as an adjuvant chemotherapy. That means here the chemotherapy is used to cure cancer, lessen the chance it will return or stop or slow its growth. That means this adjuvant chemotherapy is usually administered after the surgery, right, to kill any remaining cancer cell with the goal of reducing the chances of their recurrence. On the other hand, neoadjuvant chemotherapy, they are responsible for easing the cancer symptoms. So initially they will try to shrink the tumors that are literally causing the pain or other problems, and after that they go for surgery. Right. So here what happens? First, we are shrinking the tumor and then we are going for the therapy. So we can say that this neoadjuvant chemotherapy is delivered before the surgery. Okay. With the goal of shrinking a tumor or stopping the spread of the cancer to make surgery less invasive and more effective. Okay. So remember, this is after the surgery and this is before the surgery. So chemotherapy can be given in different ways. We can give it orally, directed to the veins, or given as shots in muscles in our arm, thighs or hips. Intraperitoneal, that is area in our body containing organs, artery, or can also be rubbed onto the skin. So here are different categories of chemotherapeutic drugs and their mode of action. So alkylating agents, which causes DNA damage but not phase specific. Platinums come under that. Anti-metabolites, DNA or RNA replication in the S phase of the cell division. Pyrimidine analogs is an example. Then anti-tumor antibiotics, isomerase inhibitors, mitotic inhibitors targeting the mitosis, etc. So some more examples are also given here. Next, immunotherapy, that is adoptive cell transfer. DC VACs, they utilize dendritic cells that has been taken from the patient's blood, stimulated in vitro with antigens from the patient's own tumor and then injected back to the patient body. So it is a type of immunotherapy in which T cells are given to a patient to help the body fight against disease. For example, cancer. So what happens in cancer therapy? T cells are taken from the patient's own blood or tumor tissue. grown in vitro under large numbers in the laboratory and then given back to the patient to help the immune system fight against cancer. In certain cases, the T cells are also changed in the laboratory to make them better able to target the patient's cancer cell and kill them. So this therapy is otherwise known as adoptive cell transfer, cellular adoptive immunotherapy, or the T cell transfer therapy. Then we have some examples of humanized monoclonal antibodies like herceptin, that is directed against HER2 receptor. So it inhibits the activation of receptor by the growth factor, thereby stimulating receptor internalization. Usually they are responsible for the stimulation of proliferation of breast cancer cells. Rituxan for the treatment of non-Hodgkin's B-cell lymphoma. So they can go and bind to a cell surface protein present on the malignant B cell, inhibiting their cell growth, inducing the cell to undergo apoptosis. Vectibix, directed against epidermal growth factor receptor, targeted for metastatic colon cancer. Arzira and Yervoy, for the treatment of chronic lymphocytic leukemia and melanoma. Arzira has got the same application as Rituxan, that is binding to CD20 on the surface of B cells, whereas Yervoy will act by blocking the CTLA4. Avastin is directed against VEGF, that means stopping angiogenesis. So here are a number of drugs, what are their targets and what is their mode of action. So we'll take one example from here, Glivec, BCR-ABL gene. So mechanism of action is tyrosine kinase inhibitor. So as we can see here, BCR-ABL gene, if it is active, ATP hydrolysis takes place, the phosphate will be transferred to the substrate protein. This is the signal for cell proliferation and survival, resulting in leukemia. But if the patient is treated with Glivec, Glivec will go and bind to the site where ATP binds. So what happens? No ATP hydrolysis, no phosphate getting activated, no signal, no leukemia. Then precision medicine in cancer treatment. So basically it is an approach to patient care that allows doctors to select treatments that are most likely to help patients based upon their genetic understanding of their disease. These are called personalized medicine. So unlike any genetic changes in cancer, epigenetic changes, as we know, that are reversible. So re-expressing the genes that had been silenced by methylation using DNA demethylating agents. We have 5-azacitidine and 5-aza-2' deoxycytidine as elective treatments for a pre-leukemic disease, that is myelodysplastic syndrome. Okay. So that's all for today's session from cancer biology. So I hope so I was able to make you understand the basic concepts regarding the cancer, the types of cancer and their application. Thank you and see you again in another session.